Fertilization creates a new organism. In this video, I am going to tell you how fertilization occurs. Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure you subscribe my channel. So today we are going to see the next video in the series of reproductive physiology, that is the fertilization. So in the previous video we have talked about the capacitation and we have seen that the sperm has reached the site of the fertilization. So now how does the ovum is going to reach the site of the fertilization? So we know that the site of the fertilization is the ampulla region of fallopian tube. And how does the ovum reach the site of the fertilization is that the translocation of ovum to ampulla of the fallopian tube. So we know that after the ovulation, what happens? The secondary oocyte with the surrounding cumulus cells is going to be released by the ovary into the peritoneal cavity, and from there the fibri of the fallopian tube is going to capture it. Now after this capture, the oocyte cumulus complex will enter into the lumen of the tube, and then by the action of the cilia, which is the fallopian tube is being lined by the ciliated epithelium. So there is cilia beating as well as the smooth muscle contraction of the fallopian tube, which will translocate the oocyte cumulus complex from fibri region of fallopian tube to the site of the fertilization, that is the ampulla of the fallopian tube. So now both sperm and egg or the ovum are at the site of the fertilization, that is the ampulla of fallopian tube. Now what is fertilization? So we all know that fertilization means that the fusion of the male and the female gamete, but it's not exactly what is fertilization. It's not the complete elaboration of the term fertilization. It is basically a multi-step process which is starting with the contact between the sperm and the egg, and ultimately it ends with the egg activation. So ultimately it results in the formation of a two cell stage, ultimately leading to the first meiotic cleavage, forming a 2-cell stage. So it is going to start the point at which the contact is occurring between the sperm and the ovum. So not only the contact but as well as a regonization happens between the sperm and the ovum, because fertilization is a species specific process. So the specific species sperm is going to fertilize only the specific species ovum. So at the time when they are contacting, there is also a regonation which is happening between the sperm and the ovum to make sure that a one particular species sperm is fertilizing or meeting with with that respective species only. So how does that happen? So if you remember the video on the structure of the ovum, where we have talked about that how does the different layers are being there which are surrounding the oocyte. So the sperm first has to pass through these layers to finally reach with the oocyte membrane. So it will pass through the cumulus matrix. It will also going to penetrate the zona pellucida via the acrosome reaction. Then the membrane fusion happens between some sperm and oocyte, and then the sperm cell content will enter into the oocyte. So the very important point to remember here is that the sperm membrane will not enter inside the oocyte. It is going to only the content of the sperm which is going to enter the oocyte. And then this is not the end of the fertilization. After that, because of the entry of the sperm, it is going to lead to the next thing, which is the activation of the egg. So what is the activation of the egg? The activation of egg means that there will be cortical granule reaction in the sperm, in the oocyte. Then there is resumption of the meiosis. So the oocyte is being arrested at the metaphase 2 of the meiosis 2. So it will complete its second meiotic division to form a oocyte which is having unduplicated haploid number of chromosomes. Then it will also eliminate the translation inhibitors, which is leading to the synthesis of the mRNA and the initiation of the embryonic deployment. and ultimately the fusion of male and female pronuclei. So you understand that we define the fertilization only by this one line, that it is a fusion of male and female gamete, but it is not only that, it is involving the contact, and in the contact also there are multiple steps. Then in activation also cortical granule reaction happens, which is going to block the polyspermy. The resumption of meiosis is occurring, then you can say the fusion is happening, and ultimately the first meiotic cleavage is going to happen, at which you will see the two cell stage, and that is the point where you are going to see a diploid cell. So now we will start with the with one by one with each step. So first is the contact. So you can see here, if you can see, this is the first step. So basically what is happening, this is a sperm which is coming in the contact with a cumulus layer. So now the sperm cell cytoplasm is having the hyaluronidase, which is going to depolymerize the hyaluronic acid, which is a cementing material keeping together the cumulus cells. So many books are saying that hyaluronidase is being released from the acrosome. Some are saying that it is present in the sperm cell cytoplasm. So it is a controversial thing, but you remember that the hyaluronidase is being released by the sperm, which is going to depolymerize or degrade the hyaluronic acid, and as a result the gap between the cumulus cells is going to be decreased, and the sperm will freely pass through the cumulus cells to finally reach this layer, which is a zona pellucida. Now after reaching the zona pellucida, the second thing happens, which is called as the sperm will penetrate the zona pellucida. So how is that going to happen? So that is called as an acrosome reaction. So what is happening, that this zona pellucida is basically a glycoprotein layer which is having many glycoproteins, ZP1, 2, 3, 4. So if you remember, we have talked during the capacitation video that during capacitation, because of the cholesterol efflux, the receptors which interact with the zona pellucida, they are going to come on the membrane of the sperm membrane. So you can see that if this is a sperm membrane, I draw the diagram to make you understand. So if this is the sperm membrane and this is the acrosome membrane, so the sperm membrane is now containing the receptors for the ZP, okay, and now because of the capacitation they have came on the outer membrane, and now this is the glycoprotein, okay. So this glycoprotein, ZP glycoprotein, so this sperm receptor will interact with this glycoprotein receptor. So this is called as a sperm ZP interaction, and this interaction, what does it happen? This is going to increase the calcium inside the sperm. So calcium levels are going to be increased inside the sperm, and ultimately what will happen? So in this, okay, so you can see in this thing what is happening, that these are this is the outer membrane of the sperm. So this is having the ZP receptors, this will interact with the ZP, and there will be interaction ultimately leading to the exocytosis of the content. So you can say acrosome is not present here. So the content will be expelled here. You can see here the content has been expelled because of the increase in the calcium level inside the sperm. So this is called exocytosis of acrosome or the acrosomal reaction. Now because of that, what happens, the acrosome is having certain enzymes, and these enzymes are going to degrade the zona pellucida. So it has degraded the zona pellucida, and it will pass through this to finally reach the next layer. You can see that this is a zona pellucida, it has degraded this layer, and now this is came, which is the egg plasma membrane. Okay, then comes the fusion of the egg and the plasma membrane. So you remember, the sperm will pass through the cumulus matrix, the sperm will penetrate the zona pellucida via the acrosome reaction, and then there is fusion of the sperm and the oocyte membrane. So it will become clear with this. So this is the egg membrane which is having the microvilli. Now this microvilli are having some blue dots, you can say. These are basically the zoono. Zoono is certain glycoprotein or protein which is interacting with a specific type of the protein present on the sperm, and they help in the fusion of the sperm and the egg membrane. And another protein which is there present on the sperm membrane is called as an ismo. So ismo also is localized on the sperm membrane or the equatorial membrane, which is going to interact with the with the egg membrane during the capacitation. So ismo and Juno are going to bind with each other, and that is how the fusion will happen. So you can see the fusion has happened, and then finally the content of the sperm. So the sperm content, such as the nucleus of the sperm, then the middle piece which is containing the mitochondria, the centriole, and the tail will enter inside the sperm. So this is how you can see here what has happened, that the sperm finally entered inside but leaving behind the sperm cell membrane, only the sperm content will enter inside the oocyte. So once it enter inside the oocyte, the next thing happens, which is the activation of the egg. So we will see it again, that the hyaluronic acid is a cementing material which is between the cumulus cells, and sperm will release a hyaluronidase which is going to degrade it, and then the receptors on the sperm will interact with the zona pellucida, which will lead to the exocytosis of the acrosomal content. And acrosome is having the enzymes such as acrosome hyaluronidase and neuraminidase, which will hydrolyze the glycoproteins of the zona pellucida, and it will penetrate it. This is called acrosome reaction. Then not only the acrosome reaction, but sperm can also do the mechanical action. So sperm can move about its neck, and it can penetrate through it. Okay, so ultimately the ismo protein which is present on the sperm membrane and the posterior acrosome membrane will interact with the zonoprotein and the CD9 complex on the oocyte microvilli, and it will lead to the fusion of sperm and the oocyte membrane. Now the sperm content, that is nucleus, mitochondria, centriole, tail, and tiny amount of cytoplasm will enter inside the oocyte, leaving behind the sperm cell membrane. All the sperm content except the sperm nucleus will be degraded inside the oocyte cytoplasm. So you can see that the mitochondria will also be degraded. Centriole will also not be degraded. So centriole and nucleus will not be degraded. Mitochondria will be degraded. So you can say that the mitochondria is of maternal origin, because the paternal mitochondria is being degraded inside the oocyte. So this completes the first part of the fertilization. Now we move on to the activation of the egg. So what does the activation of the egg is meaning? It leads that there are three things which is going to happen when the activation of egg occurs. First thing is that there is cortical granule reaction. We will see what is cortical granule reaction. Second thing, there will be resumption of meiosis. Third thing is that there will be initiation of the maternal mRNA translation and the fusion. So how does that happen? So sperm is having the phospholipase C. So when the sperm enters, the content of the sperm also enters. So phospholipase C also enters inside the egg, and it will produce the IP3, which leads to the elevated intracellular calcium levels. So this becomes clear from this very beautiful diagram, that when the sperm enters, the phospholipase C enters, it will cause the calcium release from the internal calcium stores of the oocyte, and this elevated calcium level, what is going to do? The cortical granules which are present in the oocyte, they are going to be released by the process of exocytosis. This is called cortical granule reaction, which is critical for blocking the polyspermy. So polyspermy means that multiple sperms, if they enter inside the oocyte, it's called the polyspermy. Now the gamete should be diploid. If the zygote should be diploid. If it is not diploid, it will not survive. So for that means only one sperm has to fuse one egg. So if one sperm has entered inside the egg, it will lead to the activation of the cortical granule reaction, which will block the entry of the other sperms, and it is acting as a mechanism to prevent the polyspermy. So the cortical granule, what does it do? The cortical granule is having the enzymes which is going to act on the zona pellucida. It will harden the zona pellucida, so that the sperm will not, other sperms which has binded to the zona pellucida, it can't enter inside the zona pellucida, it can't penetrate it, and it will also cleave off all the receptors you know that the sperm is binding with the zona pellucida with its receptor. So the enzymes which are released from the cortical granules, they are going to cleave those glycoproteins. So those glycoproteins, when they cleaved off, the sperm could not bind to it. So the sperms which have bonded to the zona pellucida, they can't also further progress through it, and the sperms which haven't bind to it, they also can't bind to the zona pellucida. So in both the through both this mechanism, it is going to prevent the polyspermy. Okay, the second thing which it is going to do is the resumption of meiosis. So increased levels are going to do the activation of the cell cycle. These are the details. If you can memorize it. Otherwise, a resumption of meiosis is also enough to write in the exam. So it will complete its second meiotic division to form a second polar body and a oocyte which is having haploid number of unduplicated chromosomes. The third thing is that it is going to eliminate the maternal mRNA translation, because you know that the sperm has bring only a very tiny cytoplasm. It is only the maternal oocyte which is having the cytoplasm. So there are maternal mRNAs which are present, and these are going to initiate the further embryonic development, further cleavage. Okay, so that the implantation, so that the other stages like morula formation, blastocyst formation could happen. So they should start the translation once the sperm has entered inside the oocyte. So that is also going to be eliminated because of the sperm entry, leading to the increase in the cytosolic calcium level, and then it will eliminate the inhibition of maternal mRNA translation. Then the fourth thing, or you can say which is happening simultaneously, is the fusion of the male and the female pronuclei. So how does that happening? So DNA of the sperm and the egg. So basically in the sperm and the egg, there is chromosomes which are present. So they are going to decondense to form the male and the female pronuclei. Okay, once they form the male and the female pronuclei, okay, so we will go to the diagram to understand it. So now you can see the sperm nucleus and the female and the oocyte nucleus. It is inside the oocyte. So the sperm and the oocyte DNA, they will decondense, and now you can say this is the centriole which is being derived from the sperm. So when the sperm content has entered inside the oocyte, the centriole has also entered. So this centriole will now act as a microtubule organizing center, or you can understand it will form the mitotic spindles. So these mitotic spindles will go and attach to the female pronucleus, and these two will move towards each other, because for fusion they have to move towards each other. So by the microtubules they move towards each other. When they are moving, their DNA replication is also happening. So you can understand that this is a female pronucleus which is having 23 chromosomes. This is also having 23 chromosomes. Okay, 23 chromosomes, and each of them is present like this. Okay, if this is first, second, third. Now during DNA replication, what happens? They are having 1 copies. So the number of the chromosome, it's still 23 only, but they are containing the copies now. Okay, and here also there will be DNA replication which is going to happen. So the DNA replication is happening, and then when they come together, these two membranes will fuse with each other. Okay, so after their fusion, okay, I'll delete it first. Okay, so after their fusion, what will happen? They will arrange at a mitotic spindle, and then at a metaphase plate, and then the one will go here and one will go for here. Like this, there are two cells which are going to be formed. Each of them are containing 46 chromosomes, and they are having the single copy of each chromosome. Okay, no duplicated chromosomes are being present. So this is how the first meiotic cleavage is going to happen, and you will see the two cell stage. So ideally this is what the zygote is. Okay, but during the zygote you won't see a diploid nucleus as such, but after the first meiotic cleavage only you will see a diploid nucleus. So you can understand that the fertilization is ultimately leading to the first meiotic division and the two cell stage. Okay, so with this we have completed the fertilization, starting from the sperm and the egg contact, the recognition, the sperm is passing through the cumulus, then penetrating the zona pellucida, then the fusion of the sperm and the egg membrane, then the sperm content enters inside the oocyte, leaving the sperm membrane outside. Then the calcium rise happens. Calcium rise leads to the cortical granule reaction, which blocks the polyspermy. The meiosis will be completed to form the second polar body and the oocyte with haploid number of chromosomes, unduplicated chromosomes. Then there will be initiation of the embryonic development and the fusion of the male and female pronuclei to ultimately undergoing in the first meiotic division, resulting in two cell stage. This is called fertilization. So these are the references. For further detail you can look into it. So if you like this video, please like, share and subscribe. Till then, keep learning.